Well, that was a wonderful, fun presentation, and our next presentation is going to be great too. I'm going to invite to the stage now Matt Will to introduce our next speaker. He's the director of a neuroscience behavioral lab, and he's an associate professor of psychology.
Thank you, Stephanie. It's my honor to introduce our next speaker, Dr. Paul Breslin, who earned his bachelor degree in the biological basis of behavior at the University of Pennsylvania in '87. His PhD in psychology in '91, also at the University of Penn, under the advisement of Dr. Harvey Grill. Dr. Breslin did his postdoctoral training in the laboratory of Dr. Gary Beauchamp at the Monell Chemical Senses Center in Philadelphia, the first institute created for the study of chemical senses. Following his postdoctoral training, Dr. Breslin was an assistant professor of psychology at University of Illinois in Chicago, before returning to the Monell Chemical Senses Center as an associate member for several years. In 2009, he became a graduate faculty member in the Department of Food Science and professor of nutritional sciences at Rutgers University. He still remains a very active member and principal researcher at the Monell Chemical Senses Center, while also being director of an apprenticeship program that promotes student diversity in biomedical science opportunities and training. Dr. Breslin has been extremely productive throughout his early career, publishing nearly 100 journal articles, many in the prestigious journal Nature. These prolific research efforts have been funded by multiple NIH R01 awards, as well as by numerous private foundations, including past and current project awards from the Bill and Melinda Gates Foundation Global Challenges Explorations Program. And these include a past award he received dedicated to improving global childhood nutrition, and a current award aimed at eradicating malaria. He is current editor of the journal Chemical Senses, as well as a past editorial board member of Food Quality and Preference. And for all his hard work, he has deservedly received numerous awards and honors throughout his career, including the Distinguished Scientific Award for Early Career Contribution to Psychology in the area of perception and motor performance from the American Psychological Association, also the Moskowitz-Jacobs Incorporated Award for Research Excellence in Psychophysics of Taste and Smell, which is presented annually to the outstanding junior scientists in the field of psychophysics of human taste or olfaction. And we have to thank one other person that without his help, we wouldn't have gotten Dr. Paul Breslin here, and that is his son, who after Paul had agreed to graciously come here to talk, he realized that his son's birthday was this weekend, and so his son Amos agreed to postpone his birthday party for a week so that Paul could join us here. But I think he had to promise him a pony or a motorcycle or something next week for the party. But join me in welcoming Dr. Paul Breslin.
Hi, welcome. I'm the pre-lunch speaker, and my talk is about anticipation, so I think that's appropriate. I want to thank Matt very much for inviting me, as well as Stephanie and Sarah and everyone else in the organizing committee for all the work that they've done. And because this is a food sense conference, and also because I study a little bit of nutrition, I wanted to talk a little bit about how to marry the senses with nutrition. And I gave the organizing committee a couple different titles, and they decided that I should present both of them. So the first title is Why Are the Senses Important to Digestion? And the second is Why Did Ivan Pavlov Win a Nobel? And as I hope to, over the course of the next few minutes, tell you, those are in fact the same title. So first, I want to introduce the concept of taste to you. Taste really does two things. First, taste is the way that we recognize nutrients out in the foods that we're going to eat in the world around us. This is too high. You got it? Okay. And this is how we know when food is going to be good for us. This is how we identify nutrients in the food. The other thing, which is the subject of my talk today, is I want to let you know that not only is it a way of identifying nutrients that are out there in the food we're going to eat, but it's also how our body comes to expect or anticipate nutrients coming in and has to prepare for it. So to make the first point, which I'll just do very quickly, this is a personal comment on taste loss by a woman who is a physician herself and also a patient. Now, when people have head and neck cancer and they receive radiotherapy, radiotherapy often destroys tissues that turn over and reproduce very rapidly, which is why we use it to treat cancers. But taste tissue is a tissue that turns over very rapidly all the time. So when you receive radiotherapy, you lose your sense of taste. So the best way to appreciate a sense is if you lose it, then you understand how important it is to you. And this is what she had to say in The Lancet in 1959. What is it like to lose your sense of taste? To know that the most luscious fruit is a cinder and its juice flavored with copper and bicarbonate, or that a Whitstable oyster is no more appetizing than a slug. I guess she liked her oysters. If by might of effort these cinders are forced down with copious fluid, the consequences are acute indigestion and vomiting. The patient is not hungry anyway, and it's easier to starve. So when animals, including humans, lose their sense of taste, they don't want to eat anything. You don't eat if you don't taste, because this is how you know when something is worth eating. So the impact of the senses on the body. First, I want to introduce the concept of homeostasis. Homeostasis is the idea that you have an internal environment in your body fluids, in your blood, that feeds all of your tissues with the foods and the chemicals and the nutrients that it needs to survive. And the idea was introduced by Claude Bernard in a term he called the milieu intérieur, the internal environment, and said that ideally what you want is for this to be a stable, constant environment. And he wrote, originally in French, "The constancy of the internal environment is the condition for a free and independent life. This is necessary for life." The idea introduced by Claude Bernard was picked up by Walter Cannon, who did a lot of his research at Harvard, and he wrote a book in 1932 called The Wisdom of the Body, where he expounded at length on the ideas introduced by Claude Bernard and came up with 4 principles of maintaining homeostasis. Constancy requires mechanisms that act to maintain this. Something has to be responsible for orchestrating all of this. Steady state conditions require that any tendency towards change automatically meets factors that resist change. So if you're going to put a lot of glucose into your body when you drink a Coke or a Pepsi, something has to prevent your blood glucose from going sky high. That's insulin. So these are the forces that meet that. The regulating system that determines the homeostatic state consists of a number of cooperating mechanisms acting simultaneously. So we don't just have insulin, we have dozens of things that maintain this, including hormones and all sorts of other mechanisms. And homeostasis does not occur by chance, but as a result of organized self-government. We have evolved a system for maintaining homeostasis, and it's very important. So our bodies want to maintain blood nutrient levels within a relatively constrained level. Obviously, they're going to vary. We can't help that, but we want to keep them as constant as we can, both during periods of starvation and during periods of eating. The absorption of nutrients from a large meal into the blood represents a really serious challenge to this. Our ability to maintain steady nutrient levels is difficult when you're filling your body up with food. So there's a brilliant article written by Steve Woods in Psychological Review in 1991 that was titled "The Eating Paradox: How We Tolerate Food." And what he wrote is, the act of eating, although necessary for the provision of energy, is a particularly disruptive event in a homeostatic sense. And what he means is that, and he wrote about this, and I encourage you to look up this article, it's a really great article, is that when you eat a large meal, and the larger the meal, the worse the situation. So if you sit down to Thanksgiving dinner, you're assaulting your body. There's nothing worse you can do to your body than to eat Thanksgiving dinner. Your body wants to maintain constant nutrient levels in the blood, and you're putting in a ton of salt and all this liquid and all these amino acids and all this sugar and all these fats, and they go into your blood. So how does your body maintain constant levels in the face of this assault on your body? And that's where this fellow comes in. So this is Ivan Pavlov. And he was really interested in the role of the senses in digestion. This is him, obviously, later in life, perhaps taking a nap, or maybe he blinked when the flash went off, next to one of his puppies. And he's famous for studying dogs salivating when you ring bells. Now, most of you have heard of Ivan Pavlov, and you know of him from his work on learning and conditioning. He's the father of the study of learning and conditioning. And he's perhaps most famous for that. But he actually won a Nobel for studying the digestive processes and how the senses allow us to anticipate and expect nutrients coming in. This is how your body survives Thanksgiving dinner. Your body has to anticipate and expect this assault on it of all these nutrients coming in to be able to better handle and prepare what's happening. So he wrote a work called The Work of the Digestive Glands, which is the works that were responsible for his winning the Nobel. This was followed with his work that he's known for from learning and conditioning called conditioned reflexes. And Pavlov, what his aim was to understand how the digestive reflexes might be conditioned to respond to the expectation of incoming nutrients and to prepare the body for them. The body should, of course, want to do this in order to maintain homeostasis, this idea put forward by Bernard and Cannon. So this is another picture of him with his lab assistant, actually with an animal with a duct in the side of its mouth to collect saliva from, and this is how he did a lot of his works. And he noticed the role of expectation and the senses on digestion in studies on these dogs. So they'd often have like a little salivary cannula and a gastric fistula, so they had a hole in their stomach. So he did a study published in the Works of the Digestive Glands where he took a little cube of beef and he tied a string around it. And he'd open up the dog's gastric fistula and he'd poke this piece of meat into their stomach, and then he'd close up the fistula and he'd leave the string dangling out. And he'd let it sit there for like 20 minutes. And then he'd open the fistula back up again and he'd pull on the string. And what would happen when he pulled on the string? What came out when he pulled on it? A cube of beef. Nothing. There was no digestion. Nothing happened. Absolutely nothing. So he did the next experiment, which was he opened up the fistula, put the cube of beef in there, closed up the fistula, leaving the string out, and he took a little bit of freeze-dried meat powder, and he just put it on the tongue of the dog. And the dog would start salivating and drooling. He'd wait 20 minutes, and then he'd open up the fistula and pull on the string, and what came out was nothing. It was mush. It had completely been digested. This is the role of the senses in stimulating digestion. So I like to collect stamps, and I think a mark of someone's impact on society, in this case Soviet society, is whether they have stamps or not. So these are all Soviet stamps up here. This is an Argentinian. This is a Cuban. These are from the Grenadines, and this is a Romanian stamp. And these are all stamps. There he is there in the middle there. These are all stamps of Pavlov, and I think that he has had a tremendous influence on science and society. So I'm going to get kind of technical here and talk to you about 3 experiments that I've done as a way of demonstrating the role of anticipation on the body's preparations for what's coming. So the first idea that I want to introduce to you is that our body can actually start to secrete digestive enzymes and hormones before we absorb things into the body. This is called anticipatory responses. I'm not the person who discovered this stuff. This stuff has been in the literature for about 30 years. But this is a plot of people who have simply rinsed around in their mouth and then swallowed a glucose solution, just Caro syrup, just pure glucose. And there's 2 different groups here, and I'll get into more who they are, but for this slide, it's irrelevant. And this is a plot of the change of insulin in their blood as a function of time when they rinse rinse glucose around in their mouth. And what you see is that there's a rise in insulin that happens around 9 minutes. That's before, after you swallow this, it can get absorbed into your blood. So you're secreting insulin into your blood in anticipation of incoming glucose into the blood. This is before it actually makes it into the blood. This is an anticipatory response. This is called preabsorptive insulin response. So that's one way in which this happens, and it's a very, very important mechanism for health, and I'll get more into that in a minute. Another thing that we do is we can actually, that's related to that, and I'll explain this in a minute, is that we can pre-digest food. So who here has seen the movie The Fly? So you know, in the scenes, I don't think they did this in the original, but in the later one, which was much more disgusting, there's scenes where as he's transforming from a human into a fly, the only way he can eat his food is to actually vomit digestive enzymes onto his food, and then after it's sort of partially digested, then he has to sort of slurp it back up. I know it's gross. But we actually do this all the time. When we put food in our mouth, our saliva is filled with enzymes, right? Our saliva is filled with enzymes. We start breaking food down in our mouth before we swallow it. We're just like the fly. We pre-digest our food. So the question is why? So saliva has a lot of amylase. About half the protein in saliva, and saliva is full of protein, is amylase. Amylase is an enzyme that breaks down starch. But we also secrete amylase from our pancreas. In fact, 98% of all the amylase that your body secretes comes from the pancreas. And there's more than enough that comes out of your pancreas to break down starch. More than enough. So why on earth do we do it in saliva? In fact, many animals that eat starch don't even make salivary amylase, but we do. So we make salivary amylase to break down starch in the mouth. Most amylase is produced in the pancreas, and as I just said, many starch-ingesting animals don't produce it, so this is a puzzle. What is the benefit of producing salivary amylase? And this is just to show you that within our genome, we have a wide range of numbers of copies of the salivary amylase gene. Some of you only have a couple copies of this gene in your genome. Some of you have many, many copies of this gene in your genome. And this is a separate gene from the one that makes amylase in your pancreas. You have your own gene for making amylase in saliva. You have a pancreatic amylase gene and a salivary amylase gene. The pancreatic amylase gene is the same in everybody. It's not variant. But the salivary amylase gene is highly variant. And in fact, we think that peoples who derive from cultures or groups that eat a lot of starch are the ones who have the most copies of salivary amylase genes. And the ones who tend to eat more meat and come from those kinds of cultures have fewer. So this is a very, very recent evolutionary adaptation for our species. So this is showing you that as a function of different people having different numbers of the salivary amylase gene, which is called AMY1, how much salivary amylase protein appears in their saliva. And there's a very tight relationship. The more numbers of copies of this gene you have, the more enzyme you make in your saliva. It's very, very tightly correlated. So this is a plot of dividing people into two groups. Those who make a lot of salivary amylase, called the high amylase group, HA, and those who make very little salivary amylase, the low amylase group. And what I'm plotting here is what happens to blood glucose in these people, plotted on the y-axis as a function of time, when they eat a glass full of starch. People can drink starch, it's okay. So as a control, and I'll just tell you that we did this, as a control we had these people drink a glass full of glucose. That's what that last slide was that I showed you, where you get preabsorptive insulin release in response to having glucose in the mouth. But in this case, the people are very different from each other in what happens to blood glucose. This group up here is the group that makes very little salivary amylase, and this group down here is the group that makes a whole lot of salivary amylase. So what's happening is the more salivary amylase you have, the more you're pre-digesting the starch. And oddly enough, and I think this is paradoxical, what happens is the change in blood glucose when you eat the starch is much less. So what you want is to maintain as low a profile as possible. Homeostasis ideally would be a flat line going across here. So you want your deviations from that flat line to be as small as possible. So people who make a lot of salivary amylase have less change in blood glucose because they're pre-digesting it. That doesn't make sense to me. That seems paradoxical. It seems like the more you digest it, the more quickly it should get into blood. How does that work? This is where the senses come in. What's happening down here early on is that, remember I showed you that these people are actually releasing insulin early on to glucose? So if you look at what's happening at the overall insulin profile, this is plasma insulin over time. The overall insulin profile for these groups are not significantly different to each other. But this is a plot here of change in plasma insulin very early on. And what you see is that it's only the group that makes high amylase levels that have an increase in insulin early on. And these guys really don't deviate from zero. Actually, it comes down a little bit from zero. So what's happening here is the senses are being activated. What's happening here is that when you pre-digest starch in the mouth, when you're eating starch, you break down the starch into simple sugars. The senses pick up on this, And then they can talk to the pancreas, and they can say, get ready, here it comes. You release a little bit of insulin, and that small amount of insulin that's secreted allows your body to handle the sugar when it comes in, because there's insulin already there to catch that sugar and move it into the tissues. So people who break down starch early on are better adapted to signal that sugar is going to be coming in. And this is just a plot of how much amylase that you make in saliva and the change in insulin. And you can see that the more amylase, this is not as tight a relationship as before, but it's still a significant relationship of 0.7. The more amylase you have in your saliva, the more insulin you end up secreting when you put starch in your mouth. So we differ in how much salivary amylase produce. The higher level of salivary amylase, the better we can handle starch. Regardless of our salivary amylase levels, we all eat the same amounts of starch in general. So there's two different kinds of people out there in general. There's high amylase producers and low amylase producers. But these two groups of people don't eat differently. These two groups of people eat the same Twinkies, they eat the same cupcakes, they eat the same potatoes. So this is a problem because the people who don't make much amylase are eating a lot of starch and they can't handle it very well. So this puts us at risk for hyperglycemia if you're a high starch eater and a low amylase producer. And the other thing it means is that we are taught to think of foods as being high glycemic index foods and low glycemic index foods. Don't eat that food because it'll make your blood sugar go sky high. That's a high glycemic index food. But if the glycemic index is dependent on who you are, you can't talk about a food as being high or low glycemic index. It's an interaction of the food and who you are. And so I think we need to think about that to some degree. So what role does food flavor play in anticipatory responses? So that's a direct response to the taste of the food. But most of what you eat, the familiarity you have with food, has to do with the overall flavor that you get from the food. It has to do with whether it has thyme in it, or tarragon, or cumin. So these anticipatory responses that I've been telling you about, the preabsorptive metabolic responses, are key to our physiology. So food-related sensory stimuli activate neural pathways which modulate hormonal release and enzyme activity, thereby influencing nutrient metabolism. And that's true of food flavors in general. If activation of these systems is bypassed or impaired, then alterations in nutrient metabolism occurs. And there are people who don't have these anticipatory responses, and they tend to be sick. People who are morbidly obese tend not to have these kinds of responses. If you can take people who don't have these kinds of responses and boost them, if you enhance these, then they may restore normal metabolism. Supplementation of preabsorptive insulin release in certain populations also improves their glucose tolerance. So if you can condition people to release a little bit of insulin early on in their food, they tolerate food much better. However, the specific characteristics of what stimuli do this is not known. So these are unanswered questions that I wanted to address. Is the magnitude of the response of physiological significance? Is it an all or none response, or is there a range of how people respond to these? Do differences in preabsorptive hormonal profiles influence nutrient metabolism or risk of disease? And can nutrient metabolism be altered by learning about the sensory components of foods? If you know food and you're familiar with food, you can learn about the food. And if you can learn about the food, you can know what's coming and anticipate it. So we did a study, an experiment, where we made very strange stimuli because we wanted them to be alien stimuli to people. Since we wanted to teach them what the stimuli did to them, they had to be stimuli they weren't familiar with. So we had one stimulus that was flavored lavender and eucalyptus, and it was bitter. These weren't meant to be pleasant. They were actually meant to be slightly unpleasant. So one flavor is bitter and lavender and eucalyptus. The other one was flavored with cardamom and salt. And we used these two flavoring systems as a control and an experimental flavor. And they were paired either with starch in a beverage that they drank or something that was thickened to be like starch but didn't have any calories in it at all. It was just a little bit of gum that we put in. And everyone had a flavor that was paired with calories and a flavor that was paired with nothing. So the way the study worked was we would give them their two different stimuli at the beginning and measure their response to it hormonally, how they handled these things. We would then make them drink these two different solutions in alternating days for weeks. Then they would come in for testing, and we'd measure their response to the stimuli either the way they were normally paired, so they'd get the stimulus that predicted calories, either in the presence of calories, or we'd cross them. We'd give them the stimulus that predicted calories, but we wouldn't give them the calories. And we did that for everybody, for all their stimuli. And each time they came in, we took blood from these people over a period of time. So they'd get blood taken before we started giving them the stimuli, and then we would give it to them throughout the test period. Now, everybody's response to this was a little bit different. So the easiest way to show this to you is just show you some individuals. And I just want to walk you through this. So this is one subject's response here. On the y-axis of each panel is the measure of either hormone or of blood glucose over time. And this panel is looking at insulin. This panel is looking at C-peptide. C-peptide is insulin, when it's secreted, is a pro-hormone. It's not really insulin-led, it's insulin plus. And to activate it into insulin, you cleave it. And you cleave off that little bit in the front that then allows insulin to be released. The bit that's cleaved off is called the C-peptide, and then the rest of it is insulin. So it's important to measure C-peptide because insulin is actually metabolized in the body really, really quickly. And C-peptide is not measured so quickly. So C-peptide is a more accurate measure of how much insulin your body is putting out. Down here, I'm plotting blood glucose, and over here, I'm looking at glucagon. So in general, insulin is used to push sugar out of the blood into the tissues. Glucagon does many things, but glucagon, one of the things it does with regards to blood sugar is it causes either glucose to be made de novo, or it takes glycogen in your body, and glycogen is just a form of biological starch, and it cleaves it into glucose. So you can think of it in gross terms of insulin is what makes blood glucose go down, and glucagon is what makes blood glucose go up. So if you're fasting and you're starving, your glucagon levels will be high. If you're eating a big meal, your insulin levels will be high. So the dark lines here are what happens when you're given the stimulus that predicts calories, and then you get calories. And the white dots here, what happens when you're given a stimulus that was never associated with getting calories, but you get the calories. That's the crossover. So in both cases, you're getting calories. In one case, you can anticipate them. In the other case, you can't. So what happens in the case where you anticipate them is that you release, this is before you actually are drinking the stimulus. This is when you have that cup of beverage right in front of you just as you're about to drink it, and it has that flavor coming off of it. You release a little bit of anticipatory insulin early on. And then the insulin comes on much harder and much faster earlier, and then it drops away much faster. And the C-peptide shows the same thing. In the case where you don't expect calories, it comes on much later because it's not expecting calories. All of a sudden, the calories come from nowhere. It says, I know I'm not getting calories. I'm going to chill out now. And then all of a sudden, the calories appear, and then it has to chase it. Then it has to say, oh, no, blood sugar is going sky high. I better crank insulin out. And so the insulin comes on much later. So what happens to blood glucose? What happens is that blood glucose actually is much lower in the case where you expect the calories. And in the case where you don't expect calories, blood glucose goes higher and stays up for much longer. This is looking at the same person, except in this case, this is looking at what happens when you have a stimulus where you expect the calories, or the stimulus that is you're taught predicts calories, but at the very beginning of the study before you know that, and then at the end of the study after you've learned it. So this is all the stimulus that predicts calories, but at the beginning of learning, the filled symbols are what happens at the beginning, and at the end, this is the same data I just showed you, what happens after you've learned that association. And you can see that at the beginning, the stimulus that predicted calories looked just like at the end of the study, the stimulus that never predicted calories, which makes sense because at the beginning of the stimulus, you didn't know. This is just another subject I'll show you where you got something very similar. The insulin response is lower in this person and drops off much faster. And this is the blood glucose. The blood glucose is much better controlled in this person at the end of conditioning when they can predict the calories. So in this case, when they can't predict the calories, there's a huge deviation in blood glucose, and when they can predict them, it's held much better under control. And this is the difference in the blood glucose before conditioning and after conditioning. So you can see that when you anticipate calories, your body is much better able to process the food. This is how your body handles Thanksgiving dinner, which is why if you're going to make Thanksgiving dinner for people, you have to make the same recipes that everyone is expecting. So subjects are able to lower their blood glucose depending on whether or not the starches are cued by flavors. These changes are associated with condition changes in hormonal responses, especially insulin. And these data support the view that anticipation of incoming nutrients maximizes our ability to process them and to minimize deviations in blood nutrient levels. So now I'm going to talk about nausea. Why am I talking about nausea? Well, again, this is about anticipation. So nutritionists talk about nutrients, and also there's this thing we call anti-nutrients. Anti-nutrients are toxins. Nutrients are things that help you build materials in your body and maintain tissues. Anti-nutrients are materials that don't help you build. They can actually hurt your ability to build tissues and can poison you. They are the toxins. So if your body can anticipate nutrients coming in, and just as tasting sugar can affect insulin secretion and blood sugar, if your body wants to best protect itself, then tasting anti-nutrients, and almost all toxins taste bitter. Not everything that is bitter is toxic, and not everything that is toxic tastes bitter, but most compounds that taste bitter will be toxic. That if you put these in the mouth and you taste them, if your body's going to anticipate them, there should be a biological, physiological reaction to that. And so we wanted to ask that question, and we published the study. We got the cover of Current Biology. It's a nice Flemish painting there. So the sensory properties of foods influence dietary choice. Gustatory activation also influences physiological processes that may occur in parallel to the sensory inputs. These are anticipatory responses and are hypothesized to prime the body to metabolize the ingested nutrients most effectively. That's what I just showed you happens. So now we're asking the question, since we know this happens in terms of nutrients, might it also happen for anti-nutrients? So nausea is a sensation. Nausea is a feeling, right? Nausea is like seeing red or feeling hungry. So the sensation of nausea is induced when the body identifies a consumed food as poisonous. It's a response to poisoning. Now, we can get into talking about why do you feel nauseous when you're like on a boat that's rocking up and down and you haven't eaten anything, and I can answer that if you want. But nausea connotes a very strong negative affect. It's a lot like pain. It's protective, and it teaches us don't do it again, which is why pain is there. Pain is there to tell you that wasn't a good thing. Whatever just happened, whether you did it to yourself or someone did it to you, make sure it doesn't happen again. That's why pain is there. Nausea is there for the same reason. It is your body punishing you with negative affect, saying, whatever it is you just ate, don't do that again. That was not good. So we hypothesized that oral exposure to bitter-tasting stimuli would be nauseating at high concentrations, as they would predict the potential ingestion of toxins, which are often bitter. So there's no actual poisoning going on here. You're just tasting bitter and asking what happens in response to that. And the key here is that we're talking about high concentrations because, of course, we all eat low levels of bitter taste all the time. And if you think of us over evolutionary terms as a foraging and grazing species, we have to eat things that taste bitter in order to survive. Virtually everything we eat has toxins in it. If we had no tolerance of those, we wouldn't be able to eat. In this case, I'm talking about high concentrations. So nausea is a sensation. It's subjective. So how do you measure that? Like pain and other perceptual experiences, there's no direct way to observe this. So there's only two things that we can really do here. One is we can ask people to rate their nausea, like we would ask them to rate their pain. Right? The only way a doctor can know whether you're in pain or not is to ask you if you're in pain. That's the only way they can know. There is no objective measure of it, really. And the same thing's true of nausea. But nausea is also correlated with gastric disturbances. When you feel nauseated, your stomach doesn't work properly. Normally, even as you're sitting here right now anticipating lunch, your stomach is contracting at the rate of around 3 contractions per minute. It's a very steady, normal rhythm. Your stomach has these Zen things going on all the time. It's contracting at around 3 times per minute. But when you're nauseated, it goes crazy. It starts twitching and contracting, and it contracts very quickly, and it has these very slow rhythms that come in. So you can measure that as well. So one of the things we do to measure nausea is we use this thing called a nausea profile, developed by a fellow named Muth. It's the Muth nausea profile. We call it the MNP. And we asked people, whether you felt shaky, was your stomach upset, do you feel lightheaded, do you feel sick, sweaty, queasy, ill, are you nauseous, so on and so forth. And for measuring gastric activity, you can just do this with electrodes through the surface of the stomach, just like you would measure like an EKG to measure electrical activity in the heart. You can do it around your stomach and measure electrical activity of the stomach. So the stomach has a muscle, and it has electrical activity when it contracts. And so when you measure this, you can do a power analysis on this. And so this is actually time running down this way along the y-axis and along the x-axis is frequency. And as you look across time in this kind of power analysis, as you're all sitting there, assuming you're all nice and healthy, your stomach is contracting about 3 times per minute, and that's all it's doing. So you get this nice sharp peak right at around 3 cycles per minute. And someone who's really sick, who's nauseated, when you look at them, this is what you get. You still get some activity at around 3 cycles per minute, but all hell breaks loose. The stomach is just twitching away. So when we give people something that's very bitter, what we see is a distribution of ratings of nausea like this. So what happens is some people report, even despite getting very high levels of something that's bitter, that they're just not nauseated by it. Some people are moderately nauseated, and these people out here are very seriously nauseated. So just by giving them a small volume of something that's very bitter on the tongue that they don't even swallow, these people report that they're intensely nauseated by it. We also wanted to know, did this have something to do with the intensity of it? And what we found was that for people who perceive very low intensity bitterness, and they're quite rare, they tend not to be so nauseated, but more or less, it has nothing to do with how nauseated you are, how bitter it is. As long as you perceive it as strongly bitter, you'll be nauseated by it if you have that tendency. And this is just showing that for people who are very strongly nauseated, it doesn't matter what the stimulus is. This is one stimulus that's bitter, this is another stimulus that's bitter. As long as it's bitter, regardless of what the stimulus is, it'll nauseate you if you're sensitive to nausea. And this is what's looking at activity in the stomach. This is another measure of nausea. So in people who are strongly nauseated on the scale report, when you look at their gastric activity, what you see is that they have a greatly disrupted activity of the stomach. So not only are they saying that they're nauseated, their stomach is twitching away and they're very unhappy. For the people who report that strong bitter tastes don't nauseate them, they're okay. Their stomach is just chugging along nice and it doesn't bother them. If you give them a control stimulus of giving them an intensity-matched sucrose, then what you find is that people who tend to be very strongly nauseated by bitter stimuli, they're okay with that. That doesn't bother them. So this is showing you that it's very specific to bitter tastes. They report that they're nauseated and their stomach goes crazy. So I think this is an anticipatory biological, physiological response to the anticipation that toxins are coming in. And when the stomach twitches like that, what it's doing is it's preventing itself from moving those down into the intestine to be absorbed. The stomach ceases to function normally, and it ends up trapping whatever you've eaten into the stomach. And you can, in fact, regurgitate it from that point if you so choose. So holding a strong bitter stimulus in the mouth, but not sucrose, can stimulate nausea sensations as measured by this questionnaire and by EGG measurements. Although not all subjects report that they're nauseated, that's actually true for all nauseating stimuli. So the most potent nauseating stimulus there is, and this is actually one of the things they use to train astronauts. And you can make astronauts like beg for mercy and flop on the floor if you do this to them, is you put them inside a drum that has vertical stripes in it. And you put them inside the middle of this drum, and you have them sit still, and you spin the drum around them. And if you do that at the right speed, people are not happy. And we did that to our subjects. That was our absolute positive control for nausea. And not everybody's nauseated by that. So there's a heterogeneity in the population of people who are prone to experiencing nausea and people who aren't. And that's not only true for bitter taste, it's true for really potent nauseating stimuli like this hideous rotating drum. So this supports our hypothesis and demonstrates that there are, in addition to appetitive anticipatory responses to nutrients preparing the body to digest food, aversive anticipatory responses that prepare the body to contain by preventing the stomach from releasing foods into the intestine, and repel and punish you, and you will be punished if you do this, the ingestion of anti-nutrients or toxins. So our bodies struggle to maintain healthy tissues, supplying nutrients when needed. We don't want the levels to go up too high, and we don't want them to go too low. We want them to be just so. And we struggle to do this. Ingestion is an assault on this system. It's an assault on our physiology, and we can handle the influx of nutrients best when we can prepare and anticipate them. And this is what the senses allow us to do. This is true not only in preparing for incoming nutrients, but for anti-nutrients and toxins as well. And these processes are what fascinated Ivan Pavlov and are ultimately why he was awarded a Nobel in answering my title. So I just want to conclude by pointing out that there are some areas here that are really interesting to think about pursuing in the future. So there's a lot more that can be done on how our body handles and processes sweets. Some people report, if they're regular Coke drinkers, that if they switch to a Diet Coke, they get very lightheaded. This could be that their body is actually anticipating calories and releasing insulin, and in the absence of calories being delivered as a payload in the Diet Coke or the Diet Pepsi, what happens is that the blood sugar actually drops because they're releasing insulin where there's no incoming sugar. Does anybody here ever get lightheaded when they drink Diet Cokes or get headaches? Yes, I see a couple hands going up. We can also ask the question about amino acids. We have to handle amino acids in the blood the same way as sugar, and we know almost nothing about this. When you eat a large piece of anything that has protein in it, a piece of chicken breast or eggs or souffle like we just heard in the last talk, your body has to figure out, what am I going to do with all these amino acids? They actually go into the blood. Some of them are actually broken down by the intestine, but when they go into the blood, you have to handle them, and we know almost nothing about that, so we can ask that question. Also, when we eat salt, we have all kinds of reflexes to that. So, for example, a study was published by Akishi and others that as soon as you put salt in your mouth, one of the things that happens is that your body stops making urine so rapidly. So what happens is that your body has to maintain osmotic balance. And it does that by balancing liquids and salts very, very carefully. So if it thinks salts are coming in, it has to reserve liquids to maintain osmotic balance. So it doesn't want to make pee so much. This is why people who eat a lot of salt retain a lot of fluids. And people who have congestive heart failure are told, don't eat salt. Because if you eat salt, your body's going to have to retain water to maintain osmotic balance. Another reflex that I think may happen, and this is being done by a graduate student in my lab right now, is that we have to have blood pressure changes in response to having fluids coming in and salt coming in. So I think we may actually have blood pressure reflexes just from the taste of salt that are anticipatory to these things coming in. So these are my collaborators, Kathy Perrault de Gachon, and Bob Stern, Ken Cook, and Gary Beecham were all involved in the nausea studies. Abby Mandel, Susie Alarcon, Hong Lan, and Karen Teff were all involved in the conditioning and caloric studies, and Melissa Murphy is my graduate student now working on blood pressure and salt taste. And I'll leave you with that. Thank you.
So we'll take questions now, kind of in the same format that we did last time. People stand up by the mic. Yeah, please go to the microphone. Thank you. It's for the recording purposes. Go ahead.
I grew up along the Great Lakes, and some people, as you know, get nausea from rough water, and some do not. So is there some kind of food to limit seasickness prior to going out in rough water?
So the question was about the relationship between nausea and seasickness. So I believe that nausea is a response to poisoning, and that everything that makes you nauseous is tied in with that somehow. So I think that what happens is that when you are seasick, what happens is that your body, as far as your body is concerned, is not moving. Yet the world is jumping around. So that can't really happen normally. It's not possible for the world to jump around when I'm not jumping around, except when you're on drugs. So if you take the right drugs, you can make the world do all kinds of things while you sit in your chair and watch Sesame Street. And I think that the reason moving up and down in a boat makes you sick is that your body thinks that you've been drugged, because that's really the only way it can interpret or understand you're not moving, but the world is moving around. And I think that it might be possible for you to teach your body that this isn't, that it hasn't been drugged. But for right now, the medical way of treating it is to just like turn the part of the brain that processes such things off pharmacologically so it can handle it okay. But I think that in the absence of pharmacological treatment, it should be possible because learning can actually take place there as well.
Why do we eat 3 meals a day? Why do we eat 3 meals a day instead of just grazing all day if eating is an assault on your senses?
So the question is, why do we eat 3 meals a day? So I'll be Socratic and I'll answer that with a question. So if you look at what rats do, rats are generally nocturnal. They're 180 degrees phase shifted from us. But if you look at what rats do, rats eat the same way we do. Rats eat a breakfast, some kind of lunch, and then a dinner. Usually the biggest meal of the day for them is dinner. And they either nibble throughout the day or they eat a lunch. So if you look at them when they eat breakfast, they've been fasting overnight. No one eats when they're asleep, and they sleep throughout the night. So they wake up, their system's fairly empty, and they eat breakfast. Then shortly thereafter that, they start to nibble away on lunch. And at this point, breakfast has moved out of the stomach, is now in the intestine. And its nutrients are coming into the blood, and you have all of these peptides and all of these signaling molecules indicating that you've just eaten, insulin is high, blood sugar becomes elevated, and then you move into, at that point in time, the biggest meal of the day, which is dinner. So now you're packed like a sausage. Now you have breakfast in your intestines, you have lunch in your stomach, your blood is full of nutrients, all of the signaling molecules, which signal satiety, are elevated in your body. And this is the moment when you say, I want to eat the biggest meal of the day. That's what rats do. That's what we do. This is how we behave. So the question is, why do we do that? And the reason is conditioning. The reason is that we are anticipating when we are going to need calories. So we need calories at the end of a fast because we've been fasting, and when you're fasting, you need to eat. We eat a big meal before the onset of a fast because we're anticipating the fast coming on. We say, if you sleep 4 hours a night or 15 hours a night, depending on who you are, your body says, I'm not going to be eating for half a day now. I better take calories in now in anticipation of that. And we basically are eating by the clock. So a lot of times when people eat lunch, they're not eating because they're hungry per se, they're eating because it's lunchtime. Or a lot of times people eat because food's available, right? If I put a trough of bagels out in the hallway out there, a lot of you will go to it and pick up a bagel and eat. How many people here ate a bagel this morning? How many people who ate a bagel were sincerely hungry when they ate it? Yeah, it's like 10% of you. You're not eating because you're hungry, you're eating because it's there. We eat because of the cues. We eat because of food availability. We eat because the clock says it's time to eat. This is all learning.
This is a fascinating presentation. Thank you so much. I loved what you said about the blood sugar dropping after having an artificially sweetened beverage. So would you think then that with the blood sugar dropping, a person might actually be more hungry and eat more as a result of these artificially sweetened foods?
Yeah, that's an interesting question. So pertaining to that, when you eat a meal that's low in micronutrients and high in macronutrients, the macronutrients are all basically calories, right? Your body's going to burn whatever energy it needs to burn for that moment in time, and it needs the micronutrients to survive. And everything else that isn't needed now as calories or as micronutrients is just excess calories. So you can think of anything that's going to support calories. A bagel, a piece of chicken breast, anything like that is just calories at that point in time. You don't need it. So what's going to happen is that your body's going to see this huge glut of stuff coming in, It's going to secrete a tremendous amount of insulin, and it's all going to be put into storage. It's not needed. It goes into the gas tank, wherever that may be on you. And at that moment in time, your body doesn't have access to those things. It's in the mode of storing calories out of your blood. And where does that leave you? It leaves you with, if you're eating things that are low in micronutrients, it leaves you with a lot of empty calories that you can't do anything with except store. That's being pushed out, and it leaves you hungry again. So then you have to eat again. And I think that, that doesn't directly answer your question of what happens with a diet cola, but it's about this relationship of insulin and sugar and insulin and sugar and insulin and sugar. And your body is always going to want to secrete insulin when the sugar's there and push it out. If there's no sugar there, in the case of there being a diet soda, and it makes blood glucose go too low, what happens is that when blood sugars drop and the insulin's causing you to become a little bit hypoglycemic, that will make you hungry. The reason I answered what I said first is that it's the same thing when you eat a bagel. Your body doesn't need that bagel. Stop eating those bagels. When you eat that bagel, your body doesn't need it. It just goes into storage, and then you're just hungry again. And a Diet Coke is just like an instantaneous snapshot of that.
Hi, we have a member in our psychology department that studies conditioned taste aversion, which is a very powerful, long-lasting effect. And I've always been curious why, or maybe it has, but I've just never heard of it, why people that are running diet clinics don't take advantage of that in willing participants that would say, okay, give me a conditioned taste aversion to ice cream. But then you present all these other interesting conditioning factors in our body. Do you have some ideas, or have you heard of other people using some of these conditioning associations to try and help people reduce their appetite for certain foods?
So that's an interesting and really complicated question, actually. From the conditioned taste aversion side of things, Rick Mattis did a study. So we form natural conditioned taste aversions all the time, either because we eat foods and get the flu and are nauseous and vomiting after eating a food and learn an aversion to that food because we have the flu, or because we go to frat parties and drink way too much of the punch and we alcohol poison ourselves, and then we learn an aversion to the flavor of the punch or whatever that particular booze was. And Rick Mattis did a study where he pre-selected an audience of participants for a study where they had really powerful conditioned taste aversions. And he just challenged them to eat the food that they refused to eat. And people would say, no, 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 I can't do that. And he said, no, go on, I'll pay you. I'll pay you a lot of money to do it. And they say, no, you don't understand. If I eat that food, I will vomit. And he said, I'll give you a bucket. And so he finally got these people to agree to do it. They had the bucket there, they had a wad of cash in their hand, and whatever it was that they couldn't tolerate, he'd give it to them. And the people would say, okay, here goes, and they'd put it in their mouth and go, gosh, that's not so bad. And it turns out that the memory and the association lives on in our heads for years afterwards, but we actually we don't have a distaste for those foods after a while. So they can be very long-lived if you look like weeks, months, so on afterwards, but those actually fade away in terms of our actual reactions to them, even though we remember how much we hate them and we have that fear. So there's two different processes going on there. And that has nothing to do with your question, but I don't know how clinics could use conditioned taste aversions, but I think that people could. I would worry that people would actually end up being exposed to these things and learn that they're really not so bad and that it would be overcome. The flip side of that is that Tony Sclafani has devoted much of his life to studying the reverse, which is what makes things rewarding. And the ultimate payload, the ultimate reward for anything we eat, or the most rewarding thing, is glucose in the blood. Actually, fructose doesn't do it. So high fructose corn syrup, sucrose, anything that has fructose in it doesn't deliver that payload. It's the glucose that does it. And we don't know why that is, but you can take any flavor under the sun and pair it with glucose, and animals learn to love that flavor. And if you then take the glucose away and expose them to that flavor over and over and over and over again, it doesn't extinguish. It just doesn't go away. They continue to love it. And I think that that's a particularly useful concept, because we might be able to use that that condition a non-extinguishing love for nutritious foods that we might not otherwise like if we're like, this tall and we're given Brussels sprouts.
Thank you. I'm going to throw out a hypothesis and give you a couple of reasons why I think so and ask for your reaction. So my hypothesis is that the anticipatory phase involves the endorphin pathway. And the reason for that is twofold: the emotional connection we have to food, or particular foods. And the example I used last night in Brian's talk was you associate Coors beer with ski slopes. And the other is the fact that chemo patients are given tetrahydrocannabinol pills to increase their appetite. And then the final thing is what I might call popular pharmacology, also known as the munchies.
I'm not sure where to begin with that. So there's no doubt that with reward systems, opioids are involved. But in terms of anticipation, I think that we can anticipate anything, either being positive or negative. So in the case of anticipating toxins coming in, I don't know whether opioids are involved in that. Your body actually wants to punish you under those circumstances, and opioids would be counter to that. So I think that whether opioids are coming into play in anticipation depends on what is being anticipated and whether it's useful to have opioids there. I think sometimes it wouldn't be and they wouldn't come into play. In terms of cannabinoids being involved in these systems, they are part of reward systems for sure, and they're also part of inflammatory systems, which are also extremely important in the digestive system, because one of the reasons our intestines have such incredibly powerful immune systems is that they're expecting a lot of horrible things to come into them. And the cannabinoids, I actually think, are playing a role as well in the immune system there, and which is another reason why when we eat amino acids from meat, a lot of those amino acids are actually metabolized in the gut. So when you eat glutamate, like when you eat a high MSG meal, like you eat pizza, which is probably the highest glutamate meal you can consume, 80% of the glutamate is actually utilized by the intestine, and it actually strengthens the intestine, which is, I think, why human breast milk is so incredibly high in MSG naturally. And I also think that just getting back to radiotherapy patients being given those capsules, it's not just so that they can tolerate the horribleness of it, okay, but it also helps prevent the nausea because it has anti-nausea. And I would dare I say, for political purposes, those are the lucky patients, because there are states in this union where radiotherapy patients cannot get access to THC, and if they're really nauseated, they're miserable. I think unrelenting nausea is worse than unrelenting pain. It is a horrible, horrible state to be in. And we do this to patients when we treat them all the time, and we don't have any treatment for that. And one of them is actually giving them pot or pot capsules, and states don't allow that. So that's my political spiel.
And pizza's good for you? Did you say that? Okay, sorry.
Good morning. I have a question. One of the ways that distinguishes us are blood types. And I wonder if there's a relationship between that and your high amylase and low amylase. I know when you talk about blood type and eating, people think it's hocus pocus. So I was curious about your reaction.
I am sorry to say I don't have an answer for you. I don't know what the relationship is between blood type and amylase level. There could be a relationship there, because what's known about people who tend to be high amylase or low amylase is that they have an ancestry which ties them back to groups of people who are traditionally either high starch eating or low starch eating groups of people, depending on whether they're landlocked or coastal, and that's sort of the thinking behind it. And since they're going to be potentially related to each other, they could potentially share blood groups. But I don't know if that would be causal. It could just be that they were correlated.
These are our final questions.
Pardon?
Oh, just the last two of you that will be there.
Okay. Well, I have an insight and a question. First off, my mom lost her sense of smell she was allergic to penicillin, and so as a teenager she had penicillin and lost her sense of smell. After that, well, the insight was she used immense amounts of salt in all her cooking, presumably because she could taste the salt. And that means after I had lots of salt, and after I left the house and got married, I married a woman who doesn't like the taste of salt, so my salt diet went way down. And I learned that lesser amounts, I could really taste the salt, and now my mom's cooking started getting really, really salty. But the insight was, when I was growing up, I went to the bathroom twice a day, once in the morning, once at night, sometimes in the afternoon, but usually not. And after I got married, I started having to urinate every couple hours. And I thank you for the insight that, in fact, it was the salt intake that changed, and that's why I started urinating more often. The question is, the question is, my mom ate a lot of things that she didn't like the taste of. Well, it didn't have taste to her, but she knew from when she was a teenager she used to love it, so she ate a lot of things that she used to love, and she just had the memory of it and ate them a lot. But if children are born without a sense of smell or lose it early, are there any studies on their life expectancy or their nutrition intake from the lack of smell?
So let me break that down into 3 questions for you. I'll go backwards. So in terms of being born without a sense of smell, being born without a sense of smell is rare, and it happens for a couple of different reasons. One of them is a developmental disease called Kallmann syndrome, and the other one is, I think, of more unknown etiology. And I don't know what the relationship is between sense of smell and, in terms of these disorders and life expectancy, except to say that the sense of smell is involved in many other things besides food. It's important for food, not only for knowing what food flavor is, but it's how we tell when food is spoiled. So let me shift gears here for a second. So our sense of smell declines with age. So there are people who are 95 years old out there who have a perfectly fine sense of smell, but most of them don't. On average, your sense of smell starts to turn down. I won't say take a nosedive. All right, I said it anyway, but starts to go down at around age 40, and it starts to slowly drift down. So for example, the smell of natural gas from the stove. So if you turn your stove on and you let it fill up with gas, don't do this, just an example. That thing that you're smelling is a chemical that they add to gas to make it stink. Natural gas is odorless, right? They add that there so that you can smell it, and it's a very, very hard thing to do, because not only do they have to pick a chemical that has a very unique and distinctive odor that doesn't smell like anything else in the world, it has to go away when it's burned. So when you burn the gas, there has to be no odor there. And most people who are elderly cannot smell this chemical that they add to natural gas. So they're at risk of exploding from gas leaks. And so what we need to do is find a chemical that they can smell that has that odor that goes away when it's burned, and it's a very, very difficult task. So your life expectancy is affected by your sense of smell. It's how we smell smoke and fire. It's how we smell when the tiger is behind us and the wind's blowing downwind and we're about to get pounced on. So going back to your other questions about, geez, I don't even know if I can remember them now. Oh yeah, salt, right. So a lot of times when patients lose their sense of smell, they engage in sort of compensatory enhancement of the flavor of food with other things. So a lot of the flavor that comes from a food is olfactory. Right? Flavor is the coming together of taste and olfaction and the skin senses, things that have to do with heat and tingling and all of that. And when you lose the sense of smell, you lose a lot of the dimension of what goes into flavor. And you can build things back up again by boosting that in other ways. So you find people who lose their sense of smell using more salt, they use more pepper, they use more chili pepper, they use more things that are zingy and burning, and that may be what was going on. In terms of your urination phenomena, yes, when you eat salt, you will retain water in your body, which you have to do to maintain osmotic balance. And your body doesn't want all the salt and water in it, so it gives it back to Mother Nature eventually. And it takes time to do that. So, you know, I always tell my kids not to eat a lot of salty food late in the day because you want to make it through the night. And so we avoid salty foods late in the day because it will come back out, you know, a few hours later.
Maybe I'll just enjoy my power to keep everybody from lunch here. So I'm a psychologist, and you're talking about anticipation, expectancies, which we think of as psychological terms, but I'm pretty sure you don't mean that. Is it a brain-mediated or experientially mediated thing that you get this substance on your tongue and your body starts to respond? Or is it purely done through physiological pathways, biochemical? And the situation I would want to propose is, what if you put something on somebody's tongue while they're asleep? Would you get the same effects?
So, that's a really interesting question. So just to be careful in my wording here, you distinguish between whether something's perceptual and experiential or physiological. Perception is physiology. So I wouldn't separate between those two things. So I might distinguish them between whether they're conscious or unconscious, maybe. Will you accept that as a distinction?
I guess I'm wondering, do you need a brain to have these effects you're talking about?
Wow. We want to eat lunch, okay? So, so getting back to conditioned taste aversion, for those of you who don't know what conditioned taste aversion is, if you get a flavor of anything, you can, the way they always traditionally do this in rats is they just give them some saccharin, and then they give them some drug that makes them sick, that makes them nauseous. And it's not enough to cause pain or GI distress. It actually has to be nausea that causes a conditioned taste aversion. Then they develop a distaste and a dislike for that thing. So people have shown that if you take a rat and you give it general anesthetics so that it goes to sleep like you would for going into the OR for surgery, and you put some saccharin on its tongue, and then you inject into it something that would make it nauseous were it conscious, and you wait several hours, you can rinse its mouth out, the effects of the drugs will be completely gone from it. When it wakes up, it has developed a conditioned taste aversion for that substance. As far as I know, no one has any idea how that happens, but it happens. In terms of whether you need a brain, wow. So the central nervous system is the brain and the spinal column. And all I will say, so we can go to lunch, is that in people or animals that have their spinal column cut so that they're intact but disconnected from each other, you can teach limbs associations strictly through the spinal column that don't involve the brain. So the spinal column can learn conditioning. And the nervous system of the guts has its own nervous system. So for those of you who are not physiologists, we have you know, the central nervous system and the peripheral nervous system, but there's also this third nervous system, which is the enteric nervous system. So there's a web of how all of the guts and viscera and liver and pancreas, they all talk to each other independent of anything else. They're their own little universe. And just like the spine can learn things, I'm sure that it can learn things too.
Need brains. Yeah.